In the intricate tapestry of the human body, every cell plays a crucial role. Among these, red blood cells stand out as the vital carriers of oxygen. But how are these essential cells produced? The answer lies in a remarkable protein called erythropoietin, or EPO. Erythropoietin regulates red blood cell production, a process known as erythropoiesis. Imagine a conductor leading an orchestra. EPO ensures the creation of red blood cells within the bone marrow. The body maintains a delicate balance, especially for oxygen levels. When oxygen levels dip, the kidneys release erythropoietin into the bloodstream. EPO travels to the bone marrow. It binds to receptors on red blood cell precursors, instructing them to mature. This ensures an adequate supply of red blood cells to transport oxygen and sustain life. The story of EPO's discovery is a testament to scientific curiosity and the relentless pursuit of knowledge. For centuries, the process of red blood cell production remained shrouded in mystery. It wasn't until the early 20th century that scientists began to unravel the secrets behind this vital process. In 1905, two French researchers, Paul Carnot and Clotilde de Flandre, made a groundbreaking observation. They demonstrated that injecting serum from anemic rabbits into healthy rabbits stimulated red blood cell production in the healthy animals. This finding hinted at the existence of a humoral factor, a substance circulating in the blood that regulated erythropoiesis. This elusive factor, which they termed hemopoietin, became the subject of intense research for decades to come. However, isolating and characterizing this mysterious substance proved to be a daunting task. It wasn't until the late 1970s that a significant breakthrough occurred. In 1977, a team of scientists, led by Dr. Eugene Goldwasser at the University of Chicago, successfully isolated erythropoietin from the urine of patients with severe anemia. This remarkable feat marked a turning point in our understanding of EPO. With purified EPO in hand, researchers could finally study its properties in detail. The next hurdle was to determine the structure of this crucial protein. This challenge was met in the 1980s when the amino acid sequence of EPO was deciphered. This groundbreaking work paved the way for the development of recombinant human erythropoietin, or REPO, a synthetic version of EPO that revolutionized the treatment of anemia. Erythropoietin is a complex protein intricately linked to its function. EPO's unique shape allows it to carry out its vital role in the body. EPO belongs to glycoproteins, adorned with sugar molecules. These sugars play a critical role in EPO's stability and interactions. The protein backbone of EPO consists of 165 amino acids. This chain folds into a compact globular structure. One crucial region, the receptor binding site, this site fits onto a specific receptor protein on red blood cell precursors. This binding triggers signals, instructing the cell to mature into a red blood cell. Erythropoietin's role in red blood cell production. This process, known as erythropoiesis, takes place within the bone marrow, the body's blood cell factory, and involves a series of carefully orchestrated steps. At the heart of the symphony lies a population of cells known as hematopoietic stem cells. These remarkable cells possess the unique ability to differentiate or transform into any type of blood cell, including red blood cells, white blood cells, and platelets. EPO acts as the conductor, directing these stem cells down the path to becoming red blood cells. When EPO is released from the kidneys in response to low oxygen levels, it travels through the bloodstream and binds to EPO receptors on the surface of specific hematopoietic stem cells. This binding event sets off a chain reaction within the cell, activating a series of signaling pathways that ultimately switch on genes involved in red blood cell development. Think of these signaling pathways as a complex network of messengers relaying instructions within the cell. EPO's binding to its receptor is like flipping a switch, activating these messengers and setting the wheels of red blood cell production in motion. As these cells receive EPO's instructions, they undergo a series of transformations, gradually acquiring the characteristics of mature red blood cells. They produce hemoglobin, the iron-containing protein that gives red blood cells their characteristic color and enables them to carry oxygen. Several conditions can be related to irregular erythropoietin levels, including anemia, chronic kidney disease, and certain types of cancers. Deficiencies in erythropoietin production can lead to severe anemia, while excess levels can result in conditions like polycythemia. Low levels of erythropoietin can be problematic. Symptoms may include chronic fatigue, dizziness, paleness, and shortness of breath due to insufficient red blood cell production. This is often seen in chronic kidney disease patients as the failing kidneys can no longer produce enough EPO. These symptoms result from decreased red blood cell production, leading to reduced oxygen supply to tissues. What does a high erythropoietin level mean? Elevated EPO levels can be a compensatory mechanism in response to chronic low oxygen levels in the blood, known as hypoxia. This can occur in conditions such as chronic obstructive pulmonary disease, COPD, heart failure, or living at high altitudes. However, excessively high levels can also indicate disorders like polycythemia vera, a type of blood cancer. Erythropoietin has been harnessed in medical applications, particularly in the treatment of anemia related to chronic kidney disease, chemotherapy, and major surgeries. It acts by stimulating red blood cell production, thus alleviating symptoms of anemia. Thank you for watching. Please like, share, and subscribe to our channel if this video is useful.